Well, that's kind of a fun question, because I really, before I started working here, didn't know much at all about the National Plant Germplasm System. I was ready to leave the job I had previously and was recruited to apply for this position because of the background I had, and had plant biology training, did plant physiology PhD, so I knew how to grow plants, but had been working in soybean and corn, and actually I worked a little bit with a single cell algae, so nothing to do with sunflowers or any of my crops that I have now, but it's been really interesting. How politicized agriculture is, and how much having a crop is dependent on whether people have crop insurance, whether there's approved herbicides and pesticides to use, and it's all, you know, pretty complicated. Whether the equipment they might already have from growing soybean could work for growing a brassica, which has a much round seed also, but it's much smaller. So there's all kinds of other complicated issues involved in developing new crop to be successful. Our resources have become more limited, and so that's been sort of a challenging issue that I think probably everybody faces. The overarching idea is that I really want to have collections that are that have the material available for people to use, and then what's useful to my communities. So one of the ways that I reach that then is I have worked hard to build relationships with my different commodity groups and academic groups. The way that I actually attain my goals of having available material means that I need to know what people find useful. So that's having the good relationships working relationships with people, but also then need to be able to prioritize regenerations. So probably in my different crops that I manage and maintain, they range from being 40% or even 30% to 98% available. So then how do you decide which ones to regenerate? And then also, like, can I obtain additional useful germplasm? And so just to talk a little bit about sunflower, which behind me in the cage here, wild sunflowers have been used by the breeders in the sunflower crop to incorporate useful traits such as disease resistance and tolerance to other abiotic stresses. And so obtaining and ensuring that we have the best collection of the crop wild relatives for sunflower. It's native to North America, and so all of the wild species in the genus Helianthus grow in the United States, except for one subspecies. And so we're able to ensure that we have a good geographic representation of all of the different species in the collection. We have the most comprehensive collection of wild sunflower, and it's a different story for each crop, but that's what we have for sunflower. My goals for the community is then that they are able to use the material. And so again, I reach out to people. As far as being a curator, you don't just deal with plants, growing plants here and doing regenerations. I attend a good number of meetings and meet with various scientists, have good collaborations going with different academic groups and at different universities in the United States and also in Canada and other countries to know what material they would like to have available. So, I mean, I guess to have as good a background as a person can in different aspects of plant biology. Actually being able to grow plants in the field has to be done here because we do regenerations, although some of the wild sunflowers actually don't like to grow in Iowa, and we send them out to California. So I go out there once a year to check on the stuff that we have growing out there. But being able to grow plants across the United States in the National Plant Germplasm System, there are many, many, many different species that curators maintain. So I have sunflowers and oilseed brassica and flax, among other crops, but there's sorghum, we have corn here too, there's cacao in more tropical places, there's citrus in California, lots and lots of different crops for the curators to or for an aspiring curator to research on the website, see who's got what, and just sort of dig into things. So in my oilseed brassicas, that includes, besides the genus Brassica, which includes canola and some of the close relatives there are a lot of other genera in the Brassicaceae family, and a number of those have been investigated for the possibility, the potential of new crops for biofuel and along those lines. And there are some dedicated research groups focused on those, but as far as general appreciation, that's still, sort of under development. But there's some of the Brassicaceae that are really interesting, great oil potential and characteristics in their fatty acid profiles, but it's hard to really get a new crop off the ground. A primary consideration is that you want to have good genetic or genomic representation, and without having directed markers, we use geographic representation as sort of a surrogate, which is, at least to some extent, a good way to do it. And so for sunflower, it's relatively easy to develop a collection because it's native to North America. For some of my other crops, because of the status of exchanging germplasm with other countries now is sort of in flux because of the different treaties, it's hard to actually obtain, germplasm that we don't already have as far as crop wild relatives go. Whether it's going to grow in Ames. And then I've got my backup site in California, which we've used for wild sunflowers, and then also for relatives of the genus Helianthus in the family Asteraceae. They'll grow out there. And brassicas will grow out there, although I have not had to send any of my brassica there. We've been able to manage that. We've been able to manage all the Brassicaceae here in Ames between using the greenhouse to grow them over the winter, so it's sort of a pseudo-Mediterranean climate, and then putting stuff out in the field here.